Hello, my name is Samantha Eggers, and today I will be presenting about the hantaviruses. The hantaviruses are a family of viruses that spread primarily by rodents, such as through the cotton rat, deer mouse, white-footed mouse, and many other rodents. It is spread through the aerosols from the urine, feces, and saliva of these rodents. It isn't commonly spread through a bite from a rodent. Also, it is not transmitted from human to human. The different serotypes of the viruses have specific host species. Thus, there is variation in the virus depending on the location, as the specific rodents are found in certain areas. For example, the SEOV serotype is found in the Chinese white-bellied rat, which has been known to reside in various Chinese regions, such as Beijing. Hantaviruses are uncommon in the Americas. There have been a few outbreaks that occurred in 1993, 2012, and 2017. The hantaviruses are more common in Europe and Asia. The hantaviruses in the Americas are known as the New World hantaviruses. These viruses cause general flu-like symptoms such as cough, fever, headaches, and lethargy. They may develop into hantavirus pulmonary syndrome, or HPS. This syndrome can be severe, and it has a mortality rate of about 38%. In the United States, the Cin-Nobre serotype that is found in the deer mouse, which resides in western and central parts of the United States, has caused the majority of the hantavirus cases. The hantaviruses found in Europe and Asia are known as the Old World hantaviruses. These also cause general flu-like symptoms and then may develop into hemorrhagic fever with renal syndrome. And the symptoms found with hemorrhagic fever with renal syndrome include low blood pressure, vascular leakage, and acute kidney failure. I'm going to focus my attention on the article titled Epidemic Trend and Molecular Evolution of HV Family in the Main Hantavirus Epidemic Areas from 2004 to 2016 in PR China. This article was published in 2021. These researchers were interested in discovering more about epidemiological trends of hemorrhagic fever with renal syndrome and how the gene sequences of the serotypes of the hantaviruses vary depending on their location within China. Also, they sought to discover more about the evolution of the hantaviruses. Quite a few materials and methods were utilized in this study. Demographic and epidemiological data related to hemorrhagic fever with renal syndrome was taken from the Chinese CDC reports. PubMed and the Chinese National Knowledge Infrastructure databases were used to acquire the distribution of the hantavirus genotypes. The National Center for Biotechnology Information was utilized in order to acquire the genomic sequences. Genome sequencing is accomplished through following some standard steps and using some type of sequencer. For instance, whole genome sequencing can be accomplished by following the standard steps of extracting DNA from the desired organism, purifying the DNA, shearing the DNA or cutting it into fragments, copying the DNA through polymerase chain reaction, loading the copies into a sequencer, and using a computer to analyze the sequences or order of the bases. Additionally, a number of statistical programs were used in order to analyze the data. The chi-square test was used for statistical significance. EpiInfo software was used for regional distribution. DNA Star software for genetic analysis of the hantaviruses. Mega X 6.0 for the creation of a phylogenetic tree, and Simplot software for genetic reassortment analysis. The researchers generated a lot of results from their study. Some of the results include the following. From 2004 to 2016, there were 166,975 reported cases of hemorrhagic fever with renal syndrome, with 1,689 related deaths in 31 Chinese provinces. 85% of the cases occurred in just 10 provinces. The highest number of cases occurred in 2004, decreased each year until 2009, and then started to increase in 2009. Males are more likely to become infected with a ratio of 3 to 1. 88% of the cases occurred in individuals between the ages of 15 to 64. 67% of the cases occurred in farmers. The most common host was the brown rat and the striped field mouse. Phylogenetic analysis showed about 16 serotypes of hantaviruses that are endemic in China, and the clustering pattern of M genome segments was different from that of the S segments. Taking the results into consideration and with the aim of prevention of infection, it would be wise to make the public aware of the findings. Male farmers have a high risk of becoming infected with a hantavirus due to their working environment, which increases their exposure to the feces, urine, and saliva of rodents. This population should be aware of the signs and symptoms of the infection. It has shown that earlier treatment leads to a better outcome for the infected, and it is thought that the decrease in incidence occurred due to public awareness, rodent control, and socioeconomic changes. On the contrary, it is thought that the increase in the incident rate was due to an ease in taking preventative measures, a decrease in awareness of infection, and also due to climate changes such as altered habitats and changes in population density. There are at least 16 serotypes of hantaviruses that show distinct geographic aggregation. These serotypes have specific hosts. Additionally, these serotypes differ in their cluster patterns, and the clustering pattern differences indicate the possibility of gene recombination across hantavirus serotypes. Results from this study should be considered in the research, development, and distribution of a vaccine against the hantaviruses. For instance, considering the genetic variation of the serotypes and in what hosts these serotypes are found and in what region these hosts are residing. Here you can find the references I used in order to create this presentation. Thank you very much for listening.